Hi, I'm going to talk about long-term chlorotoplasty, which shared ancestry to algal symbiosis in foraminifera. And I will start from introducing foraminifera. So foraminifera are single-cell eukaryotes. They inhabit mostly marine habitats, and they have a calcareous, that means mineral shell, and a very broad distribution. And the combination of these two allow them to be great bioindicators. That means that we can use these living beings as a tool to monitor environmental changes. And although in my lab we do a lot of stuff about biomonitoring, today I'm going to show you something a bit different, which is a unique story that we, like a discovery from the past year. So, okay. I will just want to show you here in the background, you see this stand sample that was collected from the coast of Eilat, and I just brought it to show you how foraminifera look like. So I highlighted that this is one foraminifera species, and this is another one, and there's many, many more species here, and just to show you how foraminifera looks like in their natural habitat, and also how abundant they are. And another important thing about them is that they are also called living sand, and this is because many of what we see as just grains of sand are actually foraminifera shells. Okay, so after introducing foraminifera, we can now talk about symbiosis in foraminifera. So in my study, I focus on symbiosis of these living beings, and in this symbiosis, we have two partners. One is the host, which is the foraminifera, and the second one is the symbionts, which are the algal symbionts. It's a bit similar to what you might know in corals. So the foraminifera host is it's not working. Maybe I should leave presenter view because it's stuck. Okay.
This should work.
Okay. Thanks. So the foraminifera host is heterotroph. That means that they eat others, just like us, like humans. They need to eat other plants, other animals to gain energy, while the symbionts are phototroph. That means that they can gain energy from the sun. When we combine these two, we get the holobiont system, which is a mixture of these two phototrophic system. And another important definition in this context is that we have heterotrophic hosts that harbor photosynthetic symbionts. And by doing so, we get something that is called acquired phototrophy. That means that the host is acquiring the ability to do phototrophy. And in this context, I also want to mention that symbiosis in foraminifera is not with just one type of algae. For example, in corals, they have just dinoflagellate symbionts. It's just one type of algae. In foraminifera, we have like a broad examples of symbiotic system. The first one you see in A has red algae. The one in B has green algae. And it's also beautifully appeared in how the foraminifera looks like. They have different colors. Okay, so after introducing foraminifera and symbiosis in foraminifera, I can now show you the species of foraminifera I'm going to talk about today. And I study this species that is called the Euryneidae versa. So this one is a tropical shallow water foraminifera. We collect them from the Mediterranean Sea. And before this study, there was no records of symbiosis in this species. But in the lab, we noticed that under the microscope, you can see autofluorescence of chlorophyll. So we suspicion, there should be a symbionts inside. So our first aim was to reveal what is this symbionts. And the idea was to use DNA identification, but it didn't work out very well. So we decided to try for electron microscopy, just have a look inside the cell and identify the type of the algae by how they look like. When we're talking about typical algal cell, we have a few features that we look for: the nucleus, the chloroplast, and everything that should be enclosed by some cell membrane. So this is like basic structure of algal cell. But in the micrograph that we obtained, this is what we saw. So do you see anything that you recognize here? Yeah, so we see this structures, which are chloroplasts. What we don't see the complete structure of algal cells. So although this might seem in the first sight a bit confusing, how it can be that we have just the plastids. So this is something that is well known and is called keratoplasty. So in this strategy, what the host is doing is stealing and maintaining chloroplast functionality within the host. Kerato is from the word to steal in Greek. And when we are talking about chloroplast functionality, what we are referring to is the ability to photosynthesize. So again, to gain energy from the sun. That's placed keratoplasty next to algal symbiosis under this broader definition of acquired phototrophy. So this is not unique for foraminifera. There's also other animals, other eukaryotic cells that are doing the same. For example, you have here these sea slugs that is also doing this strategy of keratoplasty. And now, after understanding what Eorina diversa is doing is keratoplasty, we can go back to our genetic approach, and we did some modification for the methods. And now I'm going to show you our results. So the results will be in the form of phylogenetic trees, which is a way to display evolutionary relationship between species. So I will start from the phylogeny of the foraminifera. So we compare the sequences of diversa to other foraminifera species, and we wanted to see which one is the closest relative to diversa. So we'll just zoom into this upper branch, and we'll see that this is our foraminifera, and this is the closest relative. So what we see here is that Eorina diversa is a sister clade, which means the most closely related to a diatom-bearing host. And then, since both of them have diatoms as symbionts, we wanted also to compare the symbionts. So, the diatom symbionts. So, the next phylogenetic analysis that we did is comparison of the foraminiferal diatom symbionts. So, we compared between diatom symbionts in different foraminifera, different colors here for the different foraminifera families, and also a few free-living diatoms. And what you can see here, we'll zoom into this branch, so you can see that these are the diatom chloroplasts that we found in Eorina diversa, and they branch within the same group of diatom symbionts of the sister foraminifera. So what we see in this point is that we have two closely related foraminifera species that also have closely related symbionts. And this was very exciting, and since we work with foraminifera, we can do more than just phylogenetic comparison. We can also look into their fossil record and see documentation of when was the first appearance of these lineages. So what you can see here is that in this table, when you look at the upper part, this is today. When you go down in the table, you go back in time, and you can see that the diatom symbionts host first appearance was where this arrow is pointing is during the Cretaceous time. So when there were still dinosaurs around on this planet, these foraminifera lineages first appeared, and they are still existing today. The same specimens that I collected are from the same lineage. And our keratoplasty species appeared in the Eocene. So we have this huge gap of time between these two lineages. So what we suggest as an explanation for this is that these two lineages share some common ancestor, and then this ancestor first learned to interact with these specific diatoms, and then in one lineage it evolved to full diatom symbiosis, and in the other lineage it evolved to keratoplasty with these diatoms. And today we see these two daughter lineages that are still present in our modern oceans. Okay, so this was the main thing I wanted to show you, and I planned for a bit more. So I have also a bit of my current work that I will skip it, so I will just wrap it up. Okay, so to conclude, what I showed you is the discovery of a new keratoplastic species, and also that these keratoplasty share evolutionary link to algal symbiosis. And the last part that I skipped is how we also show that these plastids are maintained for a long time. So we did a retention experiment to test how long they keep the plastid. So we have documentation for more than 50 days, which is considered long retention time. And to put this in the wider perspective, So when we talk about keratoplasty, it's usually placed in between these two endpoints. One is algal symbiosis, and the other point is emerging endosymbiotic event. So algal symbiosis, as I introduced in the beginning, is the interaction of a host and algal symbionts that are two separate organisms to just interact and live together. But in the case of emerging endosymbiotic event, we are talking about eukaryotic host that is engulfing these plastid source, for example, free-living cyanobacterium, and then later on in evolution, we get fully integrated plastids, leading to the evolution of modern phototrophs. And in keratoplasty, why keratoplasty is placed in the middle of this thing is because in keratoplasty, we have the host interacting usually with a specific type of algae. So the prey is selected, and therefore we need the mechanism of interaction with specific algae, just like in symbiosis. But what is happening is that the host is feeding on this algal prey, harvesting the plastid, and then interacting directly with the plastid, keeping them intact and also function in this environment. And this is why in many keratoplasty studies, they refer to these keratoplasty models as a way to study what happened in these early evolutionary stages of the evolution of phototrophs. But in our case, we show that keratoplasty can also be also represent some pathology that is closer to algal symbiosis. So this really highlights how important is it to study this keratoplasty from multiple perspectives. And thank you. I will end in this note. I would like to thank my advisor, our wonderful collaborators, and our funding sources. So thank you.
Amazing talk. I have a question about some mechanisms of the stealing. Like, can you share if you know what are the molecular mechanisms and how does the host, the foraminifera, recognizes the chloroplasts and separates them from what will be eaten?
So we know that there's a selection process because these plastids are not present in the same vacuoles that you will find food particles. So we know that there must be a selection process, but we don't know what is the process itself. And since keratoplasty is super diverse, you see it in sea slugs, which are animals, and you see it in unicellular organisms like foraminifera, and also ciliates, dinoflagellates, euglonozoa. So very, very different groups. So as this study progress, you see more and more researchers that they reveal small blocks of information about different species. But I think it's going to be very diverse process. It's not just one way to keep the plastid.
I wanted to ask whether that newly revealed closeness between symbiosis and kleptoplasty, however you pronounce that, does it necessarily exclude the resemblance to photosynthesis? It might it be that photosynthesis is also a possible result originating in the same process?
So I think, again, that since this strategy evolves multiple times in different lineages, you can also see it, for example, in dinoflagellates, when you have some dinoflagellates that are phototrophs and some dinoflagellates that are just keratoplasty species. So I think maybe in these cases you can say something like this, but I don't know if you can say it in foraminifera.
Okay, so that's the one.
Okay, so this is not the typical stuff I work on, but I thought to present stuff which are cooler and nicer to talk about. So I'm going to talk about something that has to do with keys and with time. And this is joint work with Yoav, Liron, and Moni. At the time, all at Weizmann, but Now Yoav's in the industry, and Liron is at Tel Aviv University, and Moni is a professor at Weizmann, known for cryptography and other stuff. So first crash course about cryptography. We all know and love and hear about the WhatsApp messages that we send to each other. I used it today, probably most of you or all of you. And essentially, when we send messages, we don't want to be that other people would be able to read our messages. So what do? We actually want to use some sort of secret, which is sometimes called a key, and then to mix the message up so that no one can know anything about the message that we actually send. So, and this is actually goes well beyond encryption. For example, also signatures. Let's say you go online and you want to sign a document. And I want to know that you actually is the one who signed the document. So you can use cryptography to do so in a way that no one can forge your signatures. So that's why I'm not going to have a message M throughout, but some Q, which is a query that you're going to do, which can be a message or a signature, etc. There are also pseudo-random permutations, which I'm going to talk about a bit today. So what security actually means? It means that if you get the public information of the encryption scheme, you will not get useful information on the query, let's say on the message that you sent, but also on the key, because this is the secret that you're using to generate other cryptographic instances. So what modern cryptography says is that when you look at the key, after you encrypt the message, what you get is something that should look random, because random thing tells you actually nothing. You can generate it yourself. So that's how it actually is formulated, roughly. But one of the real problems is that in real life, you do not only get the value of the encryption, because you can measure other stuff. For example, you can measure running time. You can measure the power consumption of this computer. And all these parameters give you extra information which make the crypto system possibly not secure anymore. And even though I'm doing theory, it is not only theoretical. So there's been a work, I think the most influential one is by Korsher in 1996, and he used timing attacks. So he recorded times that he took to perform certain operations in the processor to actually break RSA, which is or get secret information in RSA encryption schemes, which are like the most well-known ones. We use them every day, all the time. And actually, there has been a lot of works on this area. So many people have constructed such attacks. And actually, maybe the biggest one is this one, Spectre, which actually affected all the processors actually in the world, or most of them 5 years ago. So the main prevention techniques, there are a bit more, but it's actually fixed time algorithms, meaning let's try to run the algorithm in a fixed time fashion. So this means that we want that the running time would be independent of the query that we're going to have. Let's say independent of the message. It may be random, but not something that depends on the message or the key that we're actually trying to use. And the main drawback is that it's really hard to predict how running time behaves. Sometimes when you have an algorithm, it is random. It depends on some randomness. For example, even if you do something at your home, let's say you wash dishes, if you have many dishes, then it's going to take a lot of time. And if you have only a few dishes, it's going to take a low amount of time. And you don't want to mask the amount of time that you are going to use the sink by running each time the maximum amount. So there are a few more drawbacks. And what this work was about is trying to have new definitions that says that the running time can be benign, meaning not helpful for anyone trying to attack the system. And it is not just fixed time, so it is something else. It is a new idea. And I'm going to describe one cool construction of a pseudo-random permutation. I'm going to describe what it is, but you should think about it like an encryption, which would be secure against timing attacks. So a permutation, I'm sure you recall somewhere back in the days, is just a function which is invertible from a domain n to n. And a PRP, a pseudo-random permutation, is a random permutation that looks like a random one, but it cannot be entirely random, but it is random to a computer, to someone who tries to actually distinguish it from a random one. And you should think about it, if there is one message that I think would be good to take from this work is about the following experiment, which is of a coin flip. So if I'm taking all the great physicists that we have here, and we're going to put cameras and model how a coin is flipping in the air, then we would actually could be able to guess what the outcome is going to be after the first second the coin is in the air. We do not need to see the entire trajectory. We could give a good guess. But if we don't have this set of cameras, I'm going to let you guess this coin flip. You cannot really guess the result in a good manner. So what it means is that the randomness that you see is really dependent on the computational power that you had. So something computational really relates to randomness. So there is some interaction there. So we tackled the problems related to PRPs in small domains. So you should think of credit cards or social security numbers, and each domain size have different problems or risks that you need to address. And we build on a line of 3 works that are trying to build these permutations that should look like something random. So the first one is called the swap or not algorithm or shuffle. It is based on card shuffling. So most of this algorithm think of like this domain N as a deck of cards, and then you think about how I'm going to mix it up. And if the mixing is good, then I'm going to get something which is close to random. And what this algorithm does is actually quite simple. It takes a deck of cards. It first picks some matching. Okay, so I'm not going to describe how, but some certain matching. And for each 2 matched cards, it is going to decide via another random bit whether these 2 cards are going to be swapped or not. That's why you get the name. And what they prove is that even if you ask for 90% of the domain, let's say you ask, how do you encrypt the first credit card and the second credit card, and any 90%, you cannot really distinguish the results from a random experiment. And it takes fixed log n time. So there are good reasons to think why this can be fixed, and you can see that it is sort of uniform. You take a matching, and like I haven't told you that some cards have a different process than the other cards. But then there came a different work by Rippenstadt and Jelk, and they used something that they call the mix and cut shuffle, which essentially said, you know what, let's do this swap or not shuffle, but also do it recursively. Namely, we're going to mix the deck with the swap or not shuffle, and then the top half and the bottom half, and do it recursively. So what is it going to do? The top half and the bottom half are not completely random yet, but when we add this extra randomness to each part, we get this 90% that we had before to actually 100%, meaning after you ask the entire domain, all the credit cards, you gain no information on this secret key that we actually really care about. Okay, and then came the last construction, which is called the sometimes recursive shuffle, which did actually, again, a very natural step and a very nice idea and understanding of what's going on. And they said, you know what? 90% of the cards in the swap or not shuffle look random. So there is no need to actually mix the top half and the bottom half because the top half is already shuffled. So let's shuffle only the bottom half. And this is the new shuffle. So you do the swap or not, and then you do it in the bottom half and again and again and again. And you add some shuffling. And here you can see that this is not a uniform algorithm in the running time, meaning that some cards are going to be or some inputs are going to be computed after the first swap or not, and then we're going to be done with them. But some of them require extra mixing. So this is good in terms of running time, but we can see some problem that's going on, which is there is some correlation between the output and the running time. If the output has a lot of ones in it, meaning that it is in the bottom deck for the first few iterations, it's going to run longer time to compute it. So the computation time has correlation with the output of the encryption, and this is bad for encryption. So this gives you information. So the main idea was to actually give the same expected running time, achieve again full domain security, but not doing a padding. And the idea is extremely simple. So it is called the Janus sometimes recall. So Janus is the god from January, which we are today. So what we're going to do is to use the sometimes recursive shuffle once in the right direction and in the flip direction. And what is actually going on is that now the running time is going to depend on the inner value of these permutations, which is actually looks like a random value. It is roughly an encryption of the input. So it means that you get an encryption with an encrypted running time. Yeah, so thank you. Yeah. Yes.
So maybe I misunderstood it. What are you shuffling here?
Okay, so you can think of, right, a function, you can think of it as just a mapping from 1 up to n to 1 up to n. So we can think about it as like having n cards, which are usually ordered, and then you use some randomness to shuffle them.
So you're permuting like the unencrypted message, or like the encrypted message, or
You should think of so a pseudo-random permutation is not really an encryption, but you can use it to get encryptions. So the thing is that you just need to create something which looks random. And the idea is to use something which is
apply this to the message, to the unencrypted message.
You can apply to a message, but the idea is that once you have these cryptographic primitives, you can get many other cryptographic primitives. So like in cryptography, you can reduce some of the things to other things. Yeah, so you can get encryption from it, but you can also get just a pseudo-random permutation, something which looks random, so you can use it as well. For example, let's say you have m messages. If you're going to use a pseudo-random permutation as the message that you send, of the message that you send, then you are going to get something which looks like a good encryption scheme.
Okay, thanks.
Why couldn't you use instead of fixed time padding, just some random padding, which should essentially kill the information about?
So I'm not sure it would kill the information, because let's say if you add some random variable, then you can actually sort of with high probability remove this extra random variable that you add. So in order to really mask the true running time, you actually need to go and invest the running time of the tail. So you would still get it. Let's say I have two numbers, 0 and 100, and you want to distinguish between them. So you say, you know what, I'm going to make it hard, so I'm going to add a random variable to both of them. It must be a very distributed one, a very like, must have a very big support, because otherwise you would still distinguish between the 0 and the 100. Let's say you add a random number between 0 and 50, you will distinguish it in no time.
Okay, see.
Hi everyone, I'm Elad Zehavi. I'm currently a PhD student in the Faculty of Engineering in Bar-Ilan University under the supervision of Professor Avi Sadok. And the topic of my talk today will be fiber optic sensors. So I think we are all familiar with optical fibers, maybe because it comes out of our walls in our homes, or maybe because sometime we saw basic technicians handling optical fibers on the street. But the truth is that optical fibers are everywhere, and actually over 95% of all worldwide data is transmitted today through optical fibers. So optical fibers are everywhere, and they are here to stay, at least for the near future. And if you take all optical fibers around the world and connect them, you will reach distance of 5 billion kilometers, which actually are enough to get from Earth to Neptune. So this is a very, very long distance. So we talked until now about optical fibers from the outside, yeah? But what about how they look on the inside? So optical fibers are actually made of layers, okay? So the most inner layer is the core, and the layer after that called the cladding, and after that we have the coating and another protecting layers. But the light that's being guided inside the fiber is guided mainly in the core. And the guiding principle is called total internal reflection. And this actually said that the light that propagates inside the core is hitting the walls or the boundaries of the core, and all the information, all the light stays inside because of this principle, and this is what makes the information reach its destination. So we saw that the optical fibers are really great when it comes to data delivery, right? but I want to suggest a different approach. Actually, optical fibers can be great candidates to serve as sensors, and I'll explain why. So first of all, as you know, the light is very fast, yeah, and light transmitted inside the fiber, the data that transmitted inside the fiber can reach very high rates that can reach 100 terabits per second. Second of all, optical fibers are very cheap. If you will take 1 meter of optical fiber, it will cost you tops $1. Third of all, the propagation losses of light inside the fiber are very low, and optical fibers are very immune to electromagnetic interferences and harsh environmental conditions. So all of these things, all of these properties makes optical fibers very good candidates for sensors. And this is why it's not surprising that optical fibers sensors are actually commercially available today, mainly for temperature and strain monitoring. Okay, so one example is that optical fibers cables are integrated inside the Carmel tunnels in Haifa for temperature monitoring and to alarm on the fires, etc. And the second example is that optical fibers are also integrated in Tsingma Bridge in Hong Kong, and this is as a strain sensoring to monitor the structural health of the bridge continuously. So we saw that optical fibers actually serve today as sensors, mainly for temperature and strain. And the question is why? Why only for temperature or strain? And the question is that optical fiber designed to guide light inside the core, right? Inside the most inner layer. And the thing is that if the light propagates inside the core, so only what the light can feel, only what the light can see, it can feel, it can touch, right? So if temperature affects the core and strain affects the core, the light will be able to feel it, and then it will be able to sense it. But what about different effects? What about outer effects? Something like liquids outside the fiber. In that case, the light won't be able to sense those things simply because it cannot touch them, right, or see them. So this is the main challenge here. And to illustrate this challenge, I want to use here a slide that runs in our group for a few years now. It was created by a former group member called Chagai. And he was asking his wife, how can I explain this concept to people who are not from the field? And she said, why won't you use Lion King? Okay, so as Mufasa said to Simba, everything the light touches is our kingdom. But the problem is that the light doesn't touch much. Okay? So actually there are existing solutions that require special fibers. For example, one solution says maybe we can bring the medium we want to sense, the liquid, inside the fiber. So there are hollow core fibers, and as their name indicating, they have hollow core, and therefore we can bring the liquid inside the core of the fiber, and then we'll have a light and medium interaction, and the light will be able to tell us something about the medium. Second solution says maybe we'll bring the light outside. So in this solution, we take a fiber, and we're heating it in the middle while stretching it, and then we'll get a narrow section of fiber on the middle. This is what's called tapered fiber. And in that case, in that narrow section, some energy of the light will be outside of the fiber, and then we'll have again a medium and light interaction. So all of this nice, and it works perfectly. But the thing is that these special fibers that are not widely commercially available, or will have to make them by our own. But I want to ask a question: how can we sense outer effects like liquids with using off-the-shelf, commercially available standard optical fibers? So the solution I want to suggest relies on acoustic waves inside the fiber. And this simply say, if we cannot look outside the fiber, we can still listen. Okay, and if our fiber is blind to the outside, it is still not deaf. And I explain what does it mean. So the main idea is to use acoustic waves that propagates inside the fiber to listen to the outer medium. But the question is, how can we generate those acoustic waves in the fiber? And the solution is to send a strong light pulse inside the core of the fiber. And then for an effect that's called electrostriction, we can generate acoustic waves inside the fiber. Now, the thing is about those acoustic waves is that they are not only inside the core of the fiber, they span the entire cladding of the fiber, and they can even reach the outside of the fiber. And therefore, here we can have interaction between the acoustics and the medium outside, so we can sense it. So here we have a nice animation of what will happen when we'll put our fiber in air and generate acoustic waves. So as you can see here, we have acoustic wave packet that propagates outside to inside the cladding of the fiber. And when it hits the cladding outer boundary, it's reflected back again to the core of the fiber and goes on. This when the fiber is in air. Now we'll see what happens when the fiber is in water. So again, we have this acoustic wave packet that's propagating outward. But when it hits the boundary, we see that some of the energy is leaking outside, right? And this is when we have water outside the fiber. So we can see we have difference. Some energy is leaking outside, and maybe using the acoustical losses, we will be able to tell something about the medium outside the fiber. So we saw how can we generate acoustic waves inside the fiber, and we saw how it propagates inside the fiber, but we didn't see yet how we can sense those acoustic waves again with optics. So how can we sense those acoustic waves? The answer says we'll send another optical wave inside the core of the fiber, but now to sense the effect of those acoustic waves. So we have one optical wave, the pump. This is the strong pulse wave that's generating those acoustic waves. And then we have another wave called the probe wave, which is supposed to sense those acoustic waves. And here we can see a time trace of the probe wave when the fiber is in air. So x-axis is time, y-axis is the detected voltage of the probe wave. And as you can see here, we have different peaks that represent different reflections of the acoustic wave packet from the fiber boundary. Okay. Now, if we look closely, we can see that the distance between two peaks is 21 nanoseconds, and this is exactly the acoustic time of flight in the cladding of the fiber and backward. But now we'll see what happened when we put the fiber in water. As you can see here, now we have again the same peaks, but now each peak is lower than the previous one, right? And this is because what we saw in the animation. We saw that the acoustic wave packet is going outward to the cladding boundary, and when it hits it, some of the energy is leaking outside. So every reflection back to the core is lower, and actually by calculating the decay rate of these acoustic echoes, we can calculate what is the liquid outside the fiber. So this is the method, and I'm out of time, so I won't talk about this. I'll just sum up. So we talked about fiber optic sensors that mainly available commercially today for temperature and strain. And we talked about the challenge, how can we sense something outside the fiber if the light doesn't touch it? And we talked that we can sense it using acoustic waves, and we didn't talk about that. So this is our group members. I want to thank, in our group we are working on all the projects together, so it's really not a one-man show here. And to thank you for listening. Thank you.
Yeah, so can you use, why do you need two pulses? Can your initial pulse be like a microsecond length pulse, and then it
I
will
mean-
generate end field?
We're using pulses because there are many modes inside the I mean, if you will use like two pump waves that their frequency detuning is exactly the frequency of the acoustic mode, you will generate one acoustic mode. Okay, but if you'll use pump pulse that is because it has some time width, it has also a frequency width, right? So we'll generate all the modes in the fiber, and in that case you will have a packet of acoustic waves that propagating all together inside the fiber, and then you will get these echoes, what we saw here.
Okay.
Okay.
Yeah. Thanks.
A very general question. What other things can you sense other than like the type of liquid that's interacting with the fiber?
So in other works, we were able to sense differences between solids and liquids outside. In another work here, I wanted to show how we sensed gamma radiation effect. I can see it, never mind. Gamma radiation effect in the fiber, on the coating of the fiber, and simply by using the acoustic time of flights of acoustic waves inside the coating of the fiber, which is the third layer outside the cladding. So there is many things you can do with it.
אני רוצה להודות שוב לכל הדוברים, ותודה לכולכם שהקשבתם, שאלתם שאלות. אני רוצה שוב להודות מקרב לב לבת שבע, שמסיימת החודש את 20 שנה של ניהול מלגות אדמס. אני לא יודע מה הייתי עושה בלעדיה, פשוט ניסיון חיים. ואני רוצה לאחל לכולכם הצלחה בהמשך הדרך, שנראה אתכם פורחים, מצליחים, שנשמע עליכם, ושנשמע מהתלמידים שלכם עליכם, ושתהיה לנו שנה טובה. כל טוב.